So hello everyone. Come back to the second session, hybrid wheat and genomic selection. It's my pleasure to introduce Prof. Dr. Stephen Baenziger. And let me please to give you small words about Prof. Steve. So Dr. Stephen Baenziger um, got his BSc from Harvard University, biomedical sciences, and then he got his master degree and the PhD degree from Purdue University in the field of plant breeding and genetics. His primary research on small grain breeders, winter wheat, triticale, and barley, cultivar and germ plant development are the main goals of the program. Also developing improved breeding methodology, emphasizing biotic and abiotic stress tolerance, hybrid wheat, and the use of biotechnology are the three of his basic research. So Dr. C, please start your presentation.
Okay, well, first of all, I'd like to thank the organizers very much for inviting me. And Ahmed, I'm just seeing that there appears to be some instability in my internet, so hopefully this does not stop us.
Yeah.
I do want to talk about hybrid wheat, and I would like to acknowledge my co-authors. Many of them are former students. Vikas is a research assistant professor and the real brains behind the operation. We have lots of collaboration with other people, such as at Texas A&M. We have it with Kansas State, we have it with Simmet, we have it with the European Breeding Company. Now the topics will be a short history of hybrid wheat, the need for hybrid wheat, converting a self-pollinated crop to a cross-pollinated crop, how we're going to try to find and create heterosis, the powers of genomics, and how to make commercial scale hybrids. And then finally, what happens if we fail? Because this is probably the most high-risk research that I've ever done. Whoops. Let me see what's going on there. Okay, so a short history of hybrid wheats or hybrid small grains. I started breeding wheat in 1976. There were tremendously active programs in the late 1970s and early 80s. Most of the public programs were gone by the 90s. Now, at the same time, hybrid rice, facing similar challenges, persevered. And for 30 years, they worked trying to figure out how to make hybrids, and they could not, they were unsuccessful. And then finally, they got it to work. And so now there are 17 million hectares of hybrid rice, and there are less than 200,000 hectares of hybrid wheat. And I've often wondered in my career where we would be if the hybrid wheat programs had stayed the way the hybrid rice programs had, and had worked slowly but surely trying to make it better. Were we too impoverished of spirit or too impoverished of resources to really stick with it? Now, the reason why we got interested in hybrid wheat is that if you look at the projected gains we're going to need in wheat production from now until 2050, you need 1.4 to 1.7% annual increases in production. And currently our rate of gain is about 0.9%. So it comes down to a very simple question. If you do not have a plan to raise grain yield by 1.4 to 1.7%, you have a plan to fail. And when it comes to feeding people, failure is not an option. So just to give you a recent paper by Dick Flavelle, on this side, you can see this is the developing countries. The rate of gain for wheat is a little bit over 1%, rice is about 1%, and corn is just around 2%, or maize. If you go to the developing world, maize is a little bit less, more like 1.7, and wheat is actually at 0. And so that's what's confronting us now. We've got to do something differently so that we can make our, to meet our goals to feed the world. Now, why Nebraska? And why did we do it here? Well, historically, most of the hybrid production fields were in Nebraska. In this little red circle, which is southwest and western Nebraska, northeastern Colorado, northwest Kansas, that's where almost all of the hybrid wheat was made. It was, we're in the High Plains Desert. It's very arid. It's more conducive to cross-pollination. We have winds at 9 o'clock in the morning, which is unusual for most parts of the world. So we will, when the wheat's pollinating, we have wind to move the pollen. We have a compressed pollination window or flowering window, usually just 3 to 5 days between the earliest lines and the latest lines. And so it's much easier to have the synchrony between the male or the female. That's called the nick. Very, very simple to do that. And then we have very high irrigation and very high yields. Nebraska actually has more irrigated acres than California does. And then finally, Nebraska has a history of purchasing seed, so we would be a good market for hybrid wheat. Now, what do we propose to do? Well, the first thing you have to do is you have to change a naturally self-pollinated crop into a cross-pollinated crop. And to do that, we've now screened well over 3,000 lines between the University of Nebraska and Texas A&M University, which is our chief partner. We've done some work that shows that genomic selection should work for this trait. It's difficult to phenotype, actually not too bad to phenotype, but if you've only have 3 days to score thousands of lines is virtually impossible. And what do you do when you have adverse weather such as rain during that? We've just begun working on the female characteristics. And then long-term, looking ahead, we've spent a lot of time looking at the dominant male sterile random mating populations. And we think that may be a natural way of selecting for anther extrusion and perhaps better flowering, floral anatomy. If you've never seen what a good anther extruder looks like, the picture of the wheat head in A, you can see all the anthers are out. They've shed outside the floret. The pollen would be easily dispersed. B is an extraordinarily poor anther extruder. I'm not sure if you can see it. The anther, which is right here, is barely halfway out of the floret. Where the anther would dehisce and the pollen would shed is stuck within the lemma endopalia and fertilizing only the floret. This would be a horrible cross-pollinator or male line. Is it under genetic control? Certainly. This is an addition line from rye, and you can see rye, which is naturally outcrossing, has tremendous anther extrusion. This does extremely well also. And if you've never seen a female gape line, this is what that looks like. This slide is just to give you a little bit of an idea about the genotype by environment, in this case year, for traits related to the male side. This was work done by Nick Garst. Basically, the green are the better, the red are the worst, and the yellow are the intermediate. You can see there's a lot of G by E, but basically for anther extrusion, over the 3 years he looked, the best line stayed the best, the worst stayed the worst. If you look at anthesis date, so you're looking at your nicking date, they have the same pattern. If you're looking at height, because height has been associated with anther extrusion, we've never found that. We find our semidwarfs are extremely good. You can see that the height, again, the best are the best and the worst are the worst. And then in a year where you can have a more normal pollen duration, you can see your better lines are better and the poor are poorer. In a year where it was very hot and everything flowered at the same time, there was actually no differences among the lines for pollen duration. Anther extrusion and pollen duration are the two traits that are most critical for cross-pollination. Now, looking at the female side, and I don't know if you can see this, but where you see the red arrows, you can see the stigma extruding from the gaping anther, or from the gaping floret. The floret will normally try to self-pollinate. If it doesn't get pollinated, then the lemma and palea will split and the stigma will come out. We measured a lot of different days for that, a lot of different dates on that. And what we found was if you're looking at yield, which is the ultimate measure of cross-pollination, that basically the gape angle, when everything was fully gaping, was the best trait to look at. And if you adjust that for how good the pollinator is, that's when you get really good data. So you have to have a gape angle that's open, the stigma exerted. You have to have a pollinator that can pollinate it. And then you can get pretty good hybrid seed production. This was work by Hannah Stahl. And again, the heritabilities are pretty reasonable. Gape angle at 100%, fairly high heritable trait, so we should be able to improve that and breed it. Now that we have a mechanism for working with how to create hybrids, or at least to do the mechanical parts of hybrids, the next thing you have to do is you have to find heterosis. And in a polyploid, that may not be easy. So what have we done? We've started creating experimental hybrids and the option to look for heterotic pools. And we're using a chemical hybridizing agent, Quasar 100, and this is available, thank heavens, from Asur Plant Breeding. We have successfully made hybrids now for 6 years at the University of Nebraska and 5 years at Texas A&M. We've tested the hybrids for 5 years. Our first experiment began with a full diallele. Sounds like a kind of an archaic experiment, but I'll explain why it was so valuable for us. A full diallele would be 25 by 25, but because we plant our females in pairs, we added an extra female into this. We've now gone to the balanced incomplete mating design, and I'll explain why we changed that, where we have 50 males by 100 females. And with 700 made hybrids, we can actually estimate an additional 10,475. So again, making the experimental hybrids, this is Quasar 100. That's a 3-year supply, and that was extraordinarily valuable working with Asur Plant Breeding. This is some of the students that worked on it. This is Amanda Easterly that ran all of our yield trials. Nick worked on the male aspects. Hannah worked on the female aspects. Vikas is the brains of the operation. And we've had just a lot of great visiting scientists, other graduate students, and summer workers helping on this. If you've never seen what a commercial scale hybrid wheat breeding program looks like, this is from the Asur Plant Breeding website. And what you can see, this is their experimental lines to see if they can be sterilized. There is genetic control for the receptivity to the chemical. These white bags are to measure the sterility inside the plot. In a commercial field, they will spray the females with these long spray booms. And you can see here that it's a spray boom or a male, sprayed, male, sprayed, male. Very standard production system for producing hybrid seed. Now, when we first started using the chemical hybridizing agent, the first question was, how good was it in our hands? We figure we have roughly 28 seed per spike, and that we wanted to have at least 75% sterility. Now, ideally, you'd like to have 95% sterility, but we wanted to at least have 75% sterility, which would mean less than 25% fertile. First year we tried this, we got 5.7 seeds per head. So we were where we wanted to be, but we hoped we could do better. The next year we were better at staging, better at applying, working with this, had a lot of advice from Asur Plant Breeding. We're now down to well less than 2.6 seeds per head, which would mean that we're 90% sterile and less than 10% fertile. You can still see a few outliers. We do have a lot of males. Some of the males do not get sterilized well by the chemical hybridizing agent, but it's a pretty good system. Well, then we looked at the seed, and this really caught us off guard. This is a variety which has just normal seed from a self-pollinated wheat plant. This is the CHA seed over here, and you can see it's very shriveled. And even the CMS seed, which would be cross-pollinated also, is pretty shriveled. Now, why would that be? Well, the CHA is somewhat phytotoxic. It does harm the plant. The CMS system is less phytotoxic. But again, remember that in Nebraska, you have a very short grain filling period. So you spray a plant with a CHA, you somewhat injure it, it waits 3 days after it should have pollinated, and then opens up and gets pollinated. The CMS system waits 3 days until after it should be pollinated and opens up. And so you're probably working with a 25-day grain filling period, or maybe even shorter. So the first question that came up was, if we plant the seed that is a little bit worse or a little bit shriveled, is that a problem? Okay, so that's one of the things we had to look at. So fortunately, using a full diallele, we actually had what were self-sibs, which meant that we had the male plant had pollinated the same line as a female plant, okay? And so that's something which we had to look at very, very carefully. And what we found was we could compare the self-seed to the cross seed, which would tell us if that seed quality made an effect. And the answer was no. So we could plant our hybrid seed and know we're truly measuring the effect of the hybrid. Now, we also wondered, does it matter which way you made the cross? Can you make reciprocals? Is A by B as good as B by A? And so in that case, you wanted the biology of the plant. If it's a good anther extruder, it should be the male. If it's a poor anther extruder, it should be a female. If it's early, you want that to be the female. If it's a little bit later, you want that to be the male, because you want the female to be gaping 3 days after it pollinates when the male is pollinating. And so what we found is there were no reciprocal effects. And so yes, we can let the biology determine the way the cross is made. Now, the other part that came out of that, which was kind of interesting, was is there anywhere you might see reciprocal effects? So on average, there's no reciprocal effects, but it ended up there were roughly 85% of the time no effects, 15% of the time there appeared to be some reciprocal effects. What we don't know is if that was due to maybe perhaps poor fertilization, poor sterilization, something like that. But it looks like by and large reciprocal effects are minor. That allowed us to switch. And by the way, we're probably one of the only groups that's ever really looked at, does the CHA affect the quality of making hybrid seed? And does that affect your yield trials? We then went to a very clever procedure that our German colleagues developed called the balanced incomplete block balanced missing design. And because we have very dense markers on the parents, we were able to take 50 males and 100 females, basically 25 males from Nebraska, 25 from Texas, 50 females from Nebraska, 50 females from Texas. With 150 lines, you can make 11,175 hybrids. Now, clearly, we don't have the resources to do that. Clearly, we don't have the resources to test 11,175 hybrids. But what we did know is that if we could create 700 hybrids using estimated breeding values, we could predict another 10,400. So what we were looking at here is we had 25 males at Nebraska. We crossed each of those males to 14 out of the 100 females. Now, they always weren't to the same 14. Basically, we used a rotation so that every female out of the 100 was used 3 to 4 times. And then we did the same thing in Texas. Now, we separated those because if we lost a crossing block due to adverse weather, we didn't want to lose a year. And so we would make 700 hybrids, 350 in Nebraska, 350 in Texas. We would then test those, and then we could predict how well they would do. And even if we lost one crossing block, we can estimate the performance of 7,750. And this just gives you an idea of the power of genomics and what it can really do. Now, here is a phylogeny midpoint rooted nearest adjoining tree to look at how our lines are related. Everything in red is Nebraska, go big red. Everything in blue is Texas. And you can see that there are, among the lines, there are some clusters which are almost all Nebraska, there's some clusters which are almost all Texas, and then there's some where there's some Nebraska and some Texas. That's not surprising. We share our germplasm, so that would be expected. Everything coming out from the center that is in brown is a male. Everything that comes out in black is a female. So now you can see how we worked with these materials. In this slide, you can see the relatedness among the in silico hybrids. So this is estimating all of the relationships among all possible hybrids. Nebraska by Nebraska is red, Nebraska by Texas is green, Texas by Texas is blue. So you can kind of see how these hybrids would all fit together, their relationship, their kinship. Here is where our testing locations are. 3 in Nebraska, 3 in Texas. This is geographically quite diverse. We're in the northern Great Plains. Texas is in the southern Great Plains. In Nebraska, 524 hybrids were tested at all 3 of our locations in 2018. And of the possible 650, we were able to successfully make and test 644 hybrids. If you go to Texas, 635 hybrids were tested in Texas, and 494 were tested at all of their locations. So we made seed and we were able to do our testing of our hybrids, and 635 out of 650 were tested at least at one location in Nebraska and in Texas. So we had a really excellent and robust testing situation. Now, just a curiosity, we wanted to see, based on these hybrids, was it additive dominance or some other kind of genetic variance? And for us, basically the additive genetic variance explains most of the hybrid vigor that we're seeing, which is what you're seeing in corn also, or maize. Now, if you look at the locations in Nebraska, and if you look at the lowest yielding hybrid, the mean of the hybrids, the highest yielding hybrid, and then the best available check, you can get what we call commercial heterosis. How good are you relative to what would be out there in as a pure line? And our heterosis values range from basically -1, we had an excellent year at Alliance and the pure lines did very well, all the way up to about 8%. Now, that sounds low, but again, I want you to remember that we have no history of any kind on how to find hybrids. We have no heterotic pools, nothing like this. So this is truly random mated hybrids and how well they are doing. If you look at Texas, now we lost the McGregor site in 2019. You can see again in the same case, their heterosis averaged from 3.7 to almost 14%. But what's more intriguing, and if you look at this, now this is the 150 lines on the y-axis, and then they're clustered as a dendrogram, and then the locations are clustered by their similarities on the sort of the elevated x-axis. What you want to look at here is when you see red, that's low yielding. When you see blue, that's very high yielding, and yellow is in between. So when you start seeing lines that do a very, very well, large stretches of blue, that means you've got a very high yielding hybrid that's doing well at many, many locations. And that's exactly what you'd like to see. Now I want to come back and I want to look at our data a little bit more precisely. So this is the 2018 data, and this is from Alliance, North Platte, and Lincoln. That's all in Nebraska. And if you look at it, 9 hybrids outperformed the highest yielding inbred check. North Platte, 200 hybrids, and at Lincoln, 18 hybrids. Again, randomly made. We don't expect to find heterosis. It was by pure chance. At North Platte, though, that 200 hybrids, which would be roughly 1/3 of all the hybrids tested, were better than the best pure line, was absolutely astounding. And what it relates to is North Platte had a stressful environment, and historically hybrids are much more climate resilient, much more stress tolerant than our pure lines. And you can see at North Platte that that really bears out. Negative heterosis to a positive 15% heterosis. If you go to the next year, which was less stressful, again, Alliance, there was a very good high yielding line there. 7 random hybrids in 2019, 10 random hybrids at Lincoln in 2019. Now, if you take a look at our Texas sites, you can see again that at McGregor, 2018, 75 hybrids outperformed the best inbred check, 19 at Prosper, 18 at Bushland. Again, McGregor had a little bit of stress, so that was, again, highlighting the climate resiliency of the hybrids. They lost McGregor in 2019, but you can see at Prosper, or at Pilot Point, which is very similar to Prosper, 59 hybrids did very well. So once again, what you're seeing are really good hybrids doing really well under stressful environments, which if you think about how climate change will affect wheat production, that's a very useful trait to have. So now the question comes back is, can you actually build hybrids and can you use our predictions? And so that's the next part of our project, predicting the high yielding crosses and building the heterotic groups. And this is a slide that Vikas has just recently made. And to me, it's quite stunning. In this one, the low yielding are in blue, and that's about 4 metric tons. The high yielding are in red, and that's about 4 2/3 metric tons per hectare. This is the 700 tested hybrids and the predicted 10,475 hybrids. So in this case, you have the full prediction of every possible hybrid we could make with those 150 parents. When you go to the red quadrants, you can see this is exactly where you need to be to look for heterosis and to test for hybrids. And it's something which we could not do without our genomics at all. But it's really telling us where we want to be and how we want to work moving forward. So this is a very critical slide. 700 tested hybrids, estimating another 10,475, so that we have 11,175 hybrids that we think we should be looking at. Now, our current research is to actually make those hybrids that have been predicted to be good and to test them. And that's where we're working very strenuously right now. But this slide shows you the power of that German technique. Now, once you have heterosis, once you have ways of having good cross-pollination, how are you going to scale it up? And almost everyone believes that you can use the CHA system, but it has concerns with, can you apply it if it rains, which you can't, things like this. So in this case, they believe that the cytoplasmic male sterility system will be the major way that most people will bring product to market. Now, if you're unfamiliar with the hybrid wheat production system, it involves 3 lines. First one is an A line. It has no restorer genes and a sterile cytoplasm, and it's sterile. In order to increase the seed of that A line, you cross it by what they call an alloplasmic line. It's the exact same nucleus with a fertile cytoplasm, so it can shed pollen. When you cross this, because it's in the sterile cytoplasm with no restorer genes, it's going to be sterile. That becomes the female for the commercial hybrid production, where the R line or restorer line has restorer genes, usually in a sterile cytoplasm, mainly to prove the restorer genes are there. And when you make this cross, then you get a hybrid that has a restorer gene for the sterile cytoplasm, and it's fertile. So this is the 3-line system that most people believe they're going to be working with. There is an overlooked but really critical cross, because when you breed for A lines, you actually breed B lines and then back cross them into the sterile cytoplasm to get the A line. The difficulty with that is the B line in a fertile cytoplasm may carry genes for inhibitors of restoration, or it may carry genes for partial restoration. If it's partially restored, you can never make an A line because it's never fully sterile. If it has inhibitors of restoration, when you try to make the hybrid, you have a really hard time restoring the fertility. So you flip the cross, you take the R line, which has the restorer genes and the sterile cytoplasm, hand emasculate it, use it as the female, cross it by the B line, and you will end up with this line right here, which is identical to this hybrid in the previous slide. Now, if this hybrid is sterile, then you know you have inhibitors of restoration. If you allow it to self and you never find anything that is completely sterile, then you know you have partial restoration. So this cross is what really makes the CMS system work very, very well. Now, going back to trying to build a CMS system that works with the modern tools we have, this is work done by Boha Basnet at CIMMYT in spring wheat much faster than we can do it here. He is doing a mapping population. He's looking at a number of traits, trying to find where the RF, the restorers of fertility, that's what an RF gene is for the Timothy cytoplasm. And what he's found so far is that he found one on 1A. That's where a well-known restorer RF1 gene is. He found another major QTL on 1B, where a major gene RF3 is known to be present. Then he found one on 1D, which was previously unknown. So there are also minor restorers of fertility genes, which are hard to identify using QTLs because you have to have major differences. But he also found a previously unknown one and confirmed the presence of 2 previously known ones. This is his assay. He has taken his lines that he's mapping. He's crossed a highly fertile one and a highly sterile one to 2 different common cytoplasmic male sterile testers. This didn't restore the lower one. The upper one restored beautifully. That's how we did his assay. Now, one of the questions comes up. We were just curious as to how good cross-pollination can we get? So we have made a number of CMS lines that would be the A line, and then we were crossing them by a male line and wanted to see what the level of cross-pollination was. Now, this would be the most strict system because you can increase cross-pollination by having better synchrony. If you have the male and the female be the same line, you have the least amount of synchrony that you possibly can have, basically. But you can see that we were getting anywhere from roughly 22% up to almost 50%. And this is without really trying to optimize the system at all. So yes, you can make hybrid production seed. We can certainly get a lot better than this if we tried to optimize it, but we wanted a benchmark. This is 1 year data. We also were curious to see if the CHA caused any effects. Now, in this case, what we did is we took the CMS lines. We had some that were unsprayed, and I should be careful here because there was a little bit of when you combine harvest your CMS lines, you have to be really careful, but you almost always drag a little bit of seed from the male that surrounds the female into that plot. So in the CMS lines, there may have been a little bit of self-fertility, so you do have to watch that. And in the CHA, there would be none because you'd spray it and make it sterile. But if you have a line that's sterile already, and then you spray it with the CHA, which would just make it more sterile, the cross-pollination should give you an indication of how damaging the CHA can cause. How much injury does it? We had a common male, Ruth, which is a line that's a little bit later in a good pollen shed, and you can see that, yes, the CHA does affect the cross-pollination a little bit. Some lines are much more sensitive to it than others, but at least we now know we have an indication of how sensitive the CHA would be. Now again, if you've never seen hybrid wheat, what I want to show you here, this is the CMS with the gaping sterile. This is a partial restoration. Some florets are totally fertile, some are totally sterile. And then this is full restoration where you can see just full seed set, excellent anther extrusion. Again, those traits are heritable, so you would expect if you had a male with good anther extrusion, the hybrid should have it also. Now, we've talked a lot about production systems. The CMS, the CHA, but there are other ones which you could use, and one of those would be an inducible form of apomixis, in which case you would make the heterotic combination and then make the seed through apomixis. And this is just a paper showing that in rice, they have done some work with the genetics for asexual propagation through seeds, which is a form of apomixis. Similar research is being done in wheat, so that apomixis is a real possibility. Now, what are the outcomes of this research? Well, we want to have lines with their excellent cross-pollination capabilities, good anther extrusion or gape, and they'll be publicly available, placed in our germplasm collections. All of our hybrid protocols data will be shared in public databases and publicly available. All of our created heterotic pools and algorithms will be publicly shared, and we'll try to make sure there's no pass-through restrictions where if sometimes when you get seed, we have the right to use it, but if you share it with someone else, they have to go back to the original person. We will try to avoid that. And then, of course, our CMS markers, tester lines, and R lines will be widely available. Now, the last part I want to come up with, this is the last sort of slide I want to really sort of end on, is what happens if we fail? This is by far the highest risk research I've ever been involved in. And candidly, there are two papers that have come out. So, I mean, we have to think it could fail. I mean, we've tried hybrid research before and it didn't succeed. I think now we have the tools. The first one is strategy for selection crosses using genomic prediction. So you can use genomic prediction to predict your best crosses. But the one I like really is the one by Martin van Ginkel, where it's cross the best with the best and select the best, help and breeding selfing crops. HELP stands for Hybrid Enhanced Line Performance or Prediction. And the idea is if you can make hybrids, which we can make, we know which crosses have the highest potential yield based on the F1. The F1 is a surrogate for the lines that should come out of it. By using our genomic diversity, we will know which lines, which population should have the highest variability. So if you can estimate the F1 and the variation in its progeny, you know which crosses to make and which ones to work. You may want to use some of the modern techniques like doubled haploidy to make it more efficient and faster and things like that. But even if we fail, I think we will learn so much about how to breed self-pollinated crops that it's absolutely spectacular and an extremely valuable research. And that again comes back to this. We make 700 hybrids and we can test or we can predict 11,175. It's nothing short of spectacular. It's within the breeding program's, scope and scale to be able to do this. And so I think even if we fail, and I don't expect to fail, but even if we do, we have some really beneficial outcomes. So with that, I'm going to stop. I want to thank you. Please ask questions. I would like to thank the organizers for inviting me. And again, I'd like to highlight our collaborators. Thank you now.
Okay, thank you, Steve, for this amazing lecture. And now, if you see the published, you will find a question, so you can read it by yourself if you would like.
Okay, let me see if I could find it, because when I went to my slides, the teams sort of left. So I don't know where
I can tell the question to you. So in case of SHA, you mentioned that there is 25% fertile plants. So how do you differentiate between the F and the S plants? Do you have to check each plant manually?
Okay, that was in the first one. We did that all in bagged heads, and yes, it was done. It was done that way. It was done by bagged heads, and we do have to. We do that routinely. We don't do as many now because we're much better at it. That was our worst year, to be honest with you, and we've gotten so much better after that.
Yeah. Okay, nice. I have a question with this. So, I know that you test your hybrid weeds in more than one location. So, do you find stability in the performance? I know that G by E, there is strong G by E in the locations that you use. So, do you have stability in the hybrid weed performance for yielding?
Yes, and this is a very interesting aspect, Ahmed. Historically, the hybrid wheat is more climate resilient, and so it tends to do the best pure lines when really being well done, okay,
Yeah.
will be competitive with the best hybrids if you can grow them perfectly. But anytime there is anything which reduces the yield, anything that limits yield, the hybrids tend to stay at the top and the pure lines tend to fade away. And that's even true in rice. Many people in rice will tell you that the best hybrids will be really not much better than the best pure line under perfect conditions, but that's under imperfect conditions. And so, yes, we're seeing that the hybrids, and this is historically the case, are always at the top of the trial and they don't fall away from the top, not the way pure lines do.
Okay, thank you
so they
much,
are much
Steve.
more stable, much more stable.
Perfect. Okay, thank you. Thank you so much, Steve, for the lecture. And I think we don't have further questions to ask. So thank you again. And now we will go to the second lecture. Thank you, Steve.